Hello and welcome. Thanks for tuning in for more information on the MORS methylation donor formula created by Dr. Shane Morris. As science turns its attention to the genetics of health and cellular metabolic functions, the MTHFR, methylene tetrahydrofolate reductase test is employed to identify mutations called SNPs, single nucleotide polymorphisms, in the chromosomes of the MTHFR genes, which are associated with elevated levels of homocysteine in the blood. Homocysteine is an early warning marker of cardiovascular and heart disease. It's also associated with low levels of vitamin B6, B12, and folate, as well as kidney function, pointing to the wisdom of traditional Chinese medicine, whose practices link the kidneys and the heart into one synergistic system. MORS supports intracellular biochemical methylation processes. Methyl molecules, sometimes called CH3, serve thousands of metabolic processes, especially detoxification of unwanted chemicals and heavy metals. They serve as on-off switches of genetic chromosomal expression. The onslaught of environmental toxins coupled with poor nutritional intake of B vitamins such as folate and B12 can add up to the nutritional depletion of the very molecules that support neurotransmitters, innate purification processes, normal recovery from stress, epigenetic on-off transcriptional processes, and proper immune system function. Methyl molecules are necessary for intracellular metabolic function, epigenetic expression of health, and are foundational to the body's ability to heal itself. MORS is part of the family of systemic formula cellular healing research, and its nutrients can support the body's normal processes of recovery from stress, depression, cellular purification, adrenal support, brain neurotransmitter function, and cellular energy and longevity. Let's take a quick look at a few of the key nutritional factors in MORS. The ingredient 5, MTHF, vitamin B9. Some 35% of people have a genetic issue with the MTHFR genes that limits proper utilization of folate in the cellular metabolic process. This efficient, metabolically active form of folate supports everyone's normal methylation activities and can serve normal DNA repair activities, as well as helping the body's gene expression for its normal, healthy life processes. Methylcobalamin Vitamin B12. This is the most metabolically active form of B12 that can work directly with the cells of people with MTHFR SNPs and is particularly useful to the brain's utilization. Astragalus is an adaptogenic herb well known for its immune supportive molecules. It's an antioxidant whose polyphenols target and supports methylation processes. Pyruvate and peroxidine alpha-ketoglutarate, forms of vitamin B6, are two alpha-keto molecules that help with the metabolism of carbohydrates for energy. They help jumpstart the citric acid cycle that makes adenosine triphosphate, known as ATP energy, via cellular aerobic respiration. They support the body's normal use of fats for cellular energy. Curcumin, a well-known herb with research showing that it modulates DNA expression, supports normal immune activities, and serves cell epigenetic processes that improve methylation. It's cited to help re-educate cells to perform their normal, optimal metabolic processes. Selenomethionine is a molecule of selenium bound to methionine. It supports cellular metabolic processes such as the methionine cycle involving homocysteine and SAMe. Now, SAMe is involved in the creation, activation, and disassembly of hormones, proteins, phospholipids, as well as certain drugs. Selenomethionine supports normal DNA synthesis. Green tea, More than a powerful antioxidant that's often touted, inherent in a rich supply of polyphenols that support DNA methylation for metabolic processes, research on green tea, the Camellia sinensis, shows that it supports normal DNA repair processes. Magnesium malate, and alkalizing magnesium bound to malic acid. It supports glucose metabolic and enzymatic processes. Both magnesium and the malate play important roles in mitochondrial energy processes. Zinc is a trace mineral in short supply in the modern diet and can help the body correct MTHFR metabolic anomalies, which are often a deficiency of zinc required for DNA transcriptive processes and telomere repair. The components of MORS are much more than the application of B vitamins to circumvent the MTHFR gene mutation. It's designed to provide the body a more comprehensive and efficient array of nutrients and metabolic factors that support both cellular methylation processes and the cellular metabolic functions. Coupled with herbalomic research that demonstrates efficacious botanical influences over cellular epigenetic responses, MORS features both nutrients and herbal activation components into a synergistic matrix that supports the cell's organelles' normal functions, including self-repair activities that result from the wear and tear of daily life. MORS comes in both capsules and liquid extract. Thank you.